All right, hello everybody, welcome back. We are going to continue now in understanding how we use short tandem repeats to create a DNA profile that can be used in forensics, for example. So we're going to sort of look at this from the collection stage and all the way through processing and understanding what happens at each of these steps. So the first step is to simply collect a sample of DNA. So in forensics, for example, you might collect cells from a crime scene as evidence and then extract that DNA. So for example, you might have hair follicles, or you might, in this case, have a swab from a cheek cell. And we can use this to actually then extract the DNA from that sample in the laboratory. The next step is to use PCR to amplify those multiple short tandem repeat regions. And so what you do is you amplify multiple short tandem repeats for each PCR. And again, we were using primers to really say we're only going to be selecting in these one specific region for this short tandem repeat region. And we do this multiple times and until we get a large usable sample for PCR. The next step is something called gel electrophoresis. So now that we have these bands of multiple samples of short tandem repeats from an individual sample, we need to be able to visualize and understand the differences between them in an easy, readable way. And for this, we use a process called gel electrophoresis. And so this is just an example of what one of those gel electrophoresis look like. And essentially, it's a gel in which you can suspend each of the liquid contents of the PCR product. So you basically slot in your samples that you have, and you run electricity through this gel, and the short tandem repeats move along this gel, and but because of their length, again, we know that there's variation in these short tandem repeats between individuals. Because of their length, they're only able to move so far, where the shorter tandem repeats stop close to the original sample, or where the short tandem repeats with the longest sample don't travel very far, and the ones that have those shorter segments travel the farthest. And so basically, this is just a laboratory technique that is separating these DNA fragments by size. And then we can use this to identify similarities or differences in your samples. And so a product from a gel electrophoresis might look like this. So each of these slots are where you put your original sample. So for example, A, B, C, D, E. This sidebar is just a gauge to show us how many base pairs would extend to a specific area. So for example, if we know that our band is starting here, we know that there are approximately this many base pairs in that sample. And basically what this creates is we get these fragments of DNA that are moving through this gel across basically this electromagnetic current. You're able to get these specific banding patterns for each individual. And they're visible via fluorescence. So that's why we use this gel electrophoresis, because it's a very easy visual way to understand these differences in sizes of DNA. And these banding patterns, because again, they contain these varying lengths of DNA, correspond to a unique pattern. So each person is going to have a unique banding pattern on this. And we're able to then compare banding patterns across different individuals. And that's ultimately how we use a genetic profile in forensics and in basically in any case where you really want to know the genetic identity of a person, one person compared to another. So this is the example that your book gives, where we're looking at a forensic example, where we're looking at these, again, multiple types of short tandem repeats. And the combined pattern of all these short tandem repeats at these multiple sites is unique to a specific person. So we have, for example, our unknown saliva sample. We have the banding results for suspect A, suspect B, and suspect C. And we can see that suspect C is a perfect match because of this unique banding pattern that, again, has to do with the varying lengths of segments of these short tandem repeats. And this allows us to basically exploit DNA replication to understand the genetic profiles of an individual. So you might be wondering, okay, well, yes, we're using these short tandem repeat regions, but isn't it a possibility that multiple people will have similar short tandem repeats? What is the probability, and how, and if we're not going to be sequencing the whole genome of every person, how can we be sure that the genetic profile, that unique banding pattern we're seeing, is in fact the actual individual with whom we're interested in? And again, the short answer sort of always boils down to math. And when we actually look at this and what we use as base standard in surveillance, in forensics, and for example, disease surveillance and other biological tests that make use of PCR in this genetic profiling way, we use always 15 short tandem repeats. And that sort of gives us an idea of what percentage of people are going to share that profile. So when you only have a single short tandem repeat region, it's true, about 1 in 5 people are all going to have that same region. But if you bump the number of regions up to 15, you have something in 1 in several quintillion. So your odds are extremely, extremely low that you're going to have those same short tandem repeats. So we can say with certainty that the DNA profile is very likely correct in terms of understanding whether or not we're able to identify a unique person based on these short tandem repeat regions. So in summary for this section, first of all, DNA, we know that there's a hereditary molecule that is in all living organisms. And we know that DNA is packaged into chromosomes, and those chromosomes are located in the nucleus, at least in eukaryotic cells. We also learned that humans have 23 pairs of chromosomes in their cells, where one chromosome is inherited by the initial gene source, by two gene sources here, quote unquote, mother and father, but again, this can be whatever gene source. And then we also focus specifically on what the structure of DNA looks like. We saw that DNA is a double helix. Each strand is made of nucleotides, and the two linear strands of a DNA molecule are bound together by those complementing pairs of nucleotides, adenine, thymine, cytosine, and guanine. And we looked at that complementary pairing of DNA is really at the focus and what guides DNA replication. And we learned that PCR basically enables scientists to gain that important biological process of DNA replication and uses it to create DNA sequences and DNA profiles. Okay, that's it for DNA structure and replication. We are going to pick up um, in our next lecture. We're going to be specifically looking at mitosis and how cells actually grow. So we'll pick up there, and I will see you soon.